The general public should care about SMaHT just like they should care about any type of science that we do in the field of genomics. But SMaHT in particular is a very exciting project because it's going to look at the variation that exists within people. And that is something that has never been done before. And we will find out lots of new things that will hopefully help treat and prevent disease. And we'll also expand our base of knowledge about genomics and humans and how we evolve within ourselves and the variation that resides within us, both from birth all the way through to adulthood.
So I think SMaHT is trying to answer a really difficult scientific problem. And really answering these difficult scientific problems require people from totally different fields of science to come together and really lean on the strengths that everyone has to go about this problem head forward. And so I think in SMaHT, right, we're taking people from Mendelian disease, we're taking people from the cancer field, and we're taking everything they learned in their training, in their postdocs, in their junior faculty, all of these levels, and bringing it together to try to really bring our strengths and answer these tough problems.
For our ethical, legal, and social implications sub-study of the SMaHT network. We actually gave that study a slightly different name to make it a little bit easier to understand, and that's called I Decide. And the project is really oriented towards community engagement within the SMaHT network, really at all levels. And that strategy is something that we find is critical, and we know that the network will be much more successful in achieving the goals of this study by thinking about diversity, equity, and inclusion really at every level. And so we're using a model that we call diversity, equity, and inclusion 360, which means kind of full circle, everyone involved in the conversations related to tissue donation and research, and really trying to engage them in the study as stakeholders. So one of the questions we get asked is, how is the SMaHT network engaging stakeholders? And I'm excited to share with you that that's actually a central aim of our sub-study. We currently have two stakeholder groups. The first is something we call our Community Advisory Board. The other is a group called our Multidisciplinary Specialty Committee. Those two groups are critical. They're really important to the study because they contain individuals who have lived experience with genetic conditions, with rare conditions. They have individuals with experience themselves in either being an organ recipient or organ donor, as well as we also have in our multidisciplinary specialty group, we have geneticists and genetic counselors who have real life experience in discussing these very complex diseases that the Somatic Mosaicism project really tends to inform. These community members have been incredibly kind and incredibly engaged in being able to talk with our team around everything related to the project, whether it's the type of images that we're sharing, how the logos for the SMaHT study will look, to very critical documents such as our authorization form. Does that language make sense to this very important group of stakeholders? And so with that model, we really see the strength of the SMaHT network in that we will be creating this kind of reciprocal exchange of stakeholder input back to the team. And similarly, the project will progress to allow for us to update our stakeholders as we go. And should there be challenges or should there be successes, we want to be able to share that. And so we really see that as a best practice in really trying to diversify the perspectives that are going to be included in the SMaHT network. And I can tell you, as someone with a background in health equity and in really understanding the need to make this science more accessible, we really think that this model of the DEI 360 is going to be quite important and quite helpful. We're already seeing that. Actually, we've convened our first few CAB sessions, and it's been really incredible to get that feedback. So we're very excited about that.
And so one of, I think, the big benefits of how the SMaHT network was designed is that it both includes data production aspects as well as really innovative aspects as part of what the TTDs are doing within the SMaHT network. And over the last year, it's been really exciting to see a lot of the development on both sides in terms of some of the new discoveries as well as new understandings of how we approach this question of somatic variation. I think some of the really exciting stuff has been thinking about how do you get more accuracy when actually looking at somatic variants. This includes both developing new tools for having higher accuracy sequencing so you have much more confidence when you actually identify a variant that it actually is something that's real as opposed to a false positive. And similarly, how do you leverage additional technologies to have more certainty with any of the somatic variants you find? I think when it comes to what the SMaHT network is trying to accomplish, really one of the things we're really emphasize is, can we produce a really high quality map of where somatic variants are? And this is essential to really have these tools in place that give us confidence that when we actually do identify a somatic variant, that it really exists. I think another big part of the SMaHT, which I'm very excited about in terms of thinking about really new discoveries over the last year, is better understanding how somatic variation is existing within some of the more challenging portions of the human genome. And these are areas that traditionally have actually been understudied from somatic variation because of the challenges associated with sequencing these areas. We know that these areas are actually involved in many different diseases, and so it really is imperative for us to have an understanding of how somatic variation within these areas exists and what the degree to which that exists. And I think that some of the advances that we're seeing within long-read sequencing, as well as in actually assembling genomes for each of the individuals and using that as a tool for mapping reads, is really promising in terms of opening up these new portions of the genome in terms of our understanding of the role of somatic variation in normal health as well as disease.
So how are donors' identities being protected even though data are public? So for the SMaHT Biobank, being good stewards of the donated gifts that donors and their families are providing for the program, it is our responsibility to make sure that donor privacy and confidentiality is of the utmost importance. So there are safeguards in place to protect donor information. One of those is that anything that identifies the donor, including names, date of birth, addresses, anything medical record related, is not provided to the SMaHT investigators. It is also not stored in the database where data will be collected. So that's the first safeguard. Second, investigators are given no information about donors or their families. Even the tissues that they will be using for their analysis are only labeled with a code number. And then also investigators need to sign an agreement that the policy working group formulated that they will never try to identify the donors or their families.
So I'm going to talk about how understanding somatic mosaicism can contribute to our knowledge of human development and disease susceptibility and therapeutic interventions. We have several cells, and then if we can mark certain cell into certain color, and then we can see how those cells behave. So that is pretty much similar when we study about the somatic mosaicism. So somatic mosaicism is we define specific variant, and then we're gonna take advantage of those variant as different painting color. So we can see how cell behave, and then how cell goes somewhere, and then which organ they want to, and then they contribute. So because of the many scientific community use the transgenic mouse model, but however, we cannot make transgenic human, because that is a highly illegal as well as unethical. So now we're gonna use the somatic variant, which is very physiologic event, and then we're gonna take advantage of that and then to infer what's going on and how we can just making those variant as some kind of marker for the tracing the human cells without any ethical issues.
And my answer is because it is a source of human genetic variation, changes in DNA, that is almost completely ignored. We've been sequencing people for a long time, and we have been making the assumption that the DNA that we sequence when we get a blood sample from them or a saliva sample, we make the assumption that the DNA in those cells is the same as in every other cell in their body. But we know that that's not true. And it's my belief that this vast, under-ascertained source of human genetic variation may be contributing a lot to human normal development and even human disease. So I'm really excited that we are looking for these hidden types of genetic variation that I think are going to end up playing a big role in human health and disease.
We actually have an outreach program that we have set up. We are actually planning a lot of the outreach programs for the researchers outside of the network, including the workshop. We are actually generating a lot of the benchmarking data for people to use, which are open access. And so we are planning to hold the workshop in order to teach the researchers about the best practices or the tools that we are developing the SMaHT Network. We also have public-facing websites that's coming up, and so hopefully that will be very helpful, containing educational material for the general public to know the importance of the research about somatic mosaicism, how that can actually impact the general public in terms of their health and disease, as well as our understanding of genome in general and how that changes over time as we age.
And what excites me most about SMaHT is the fact that we're building these new tools and technologies to analyze data on somatic mutations. And this really allows us to get unprecedented insights into how our tissues are acquiring somatic mutations. And I think what's great about the consortium is that we're coming together as a community to really build and enhance these tools to make them the best they can possibly be.
So I love that question, actually. Somatic mutations, which are present in some cells, but not all cells, are familiar because they are drivers of cancers. And cancers, driven by these somatic mutations, overgrow all the normal cells of the body. And so it's very easy to find those somatic mutations. You take out the cancer and somatic mutations all over it, okay? But now we're just beginning to realize that somatic mutations are important for all kinds of conditions. For example, focal epilepsy, some kinds of autism spectrum disorders, some forms of schizophrenia, but that they're ubiquitous in tissues that appear normal to the naked eye. And the disease-associated somatic mutations can be present in a tiny fraction of cells, just in that one tissue. And so we don't yet know what are the best ways to find them. How do we discover them? That's one big problem. Is this question of technology. And there's a big piece of SMaHT that's going to really contribute to that, really work that out. We've got multiple different centers. We don't know if you just take regular old sequencing methods and just do it really, really deeply, or whether you want to do long-range sequencing, or whether you want to do targeted sequencing. And that's what we're going to find out. We have multiple centers trying all these different methods, and then we're going to benchmark them. We're going to A/B them. We're going to compare our notes, and we're going to figure out what are the strengths and weaknesses of all of these different methods, and what is the most cost-effective way to find out what we want to find out about somatic mutation in normal appearing tissues, and then we can apply that to disease-associated tissues. And then secondly, we don't know what we're going to find when we start exploring the universe of somatic mutations that lies within each of us. You know, once we actually dip down below the surface and explore this tremendous genomic diversity that occurs at the cellular level in each of us, what's normal? And again, that's what we're going to understand when we analyze normal tissues from a large number of individuals. We can get a sense of what the range of variation is, and what patterns we can expect from disease-associated somatic mutations. So it's that benchmarking aspect of it that's going to be so important and that's going to have such important applications to patients and healthcare practices.
Mutations, they're like birthmarks for our cells. So every time a new cell is being created, it's almost always has a birthmark. And typically they don't do anything. But these birthmarks are being inherited to next progenies, daughters of the cell. And we can use this to reconstruct histories of the cells in our own body. So we can learn about ourselves when we were even not born. So we have now a way to trace it. And moreover, we can actually learn something about our development. That excites me a lot about somatic mosaicism.
So the primary objectives of the SMaHT network are to uncover the underlying biology created by somatic mutations that occur throughout the different cells in our body. Much is unknown about these somatic mutations and how they contribute to health, disease, and across the lifespan. It's known that there are mutations that occur in somatic cells that do contribute to disease, but we really don't understand how they occur, if there are specific regions in the genome where they are more prevalent. Specific tissues have different predisposition to different types of mutations. And so we're hoping through the SMaHT network to uncover the biases that occur potentially in different cell types, in different regions of the genome, and how they occur across the lifespan. The outcome and goals of the program are to develop a somatic catalog of all these types of mutations over a cohort of 150 people and about 20 tissues per person. And additionally, what this program is also going to do is drive technology development. So through the SMaHT network, we're funding studies that will increase our ability to detect mutations on a much smaller scale, so in increased mutation rates, so we can really understand how small numbers of mutations can contribute to disease. So mutations in small numbers of cells actually are contributing to these phenotypes. And lastly, we're really looking to create a workbench where anybody in the research community can play with the data and understand how these different types of mosaicism and these different types of mutations are interacting in their favorite study and their favorite gene.
As our cells divide during our life, they each acquire mutations, which are changes in their DNA. That starts when we are conceived and keeps going throughout our entire life. And so every cell in our body has a unique set of mutations. And what's fascinating about that is that we all contain multitudes. Each of our cells have their unique genetic profile, which as we age can lead to diseases and cancer, but is a natural part of life.
So I was asked to talk about somatic mutations and how somatic mosaicism can contribute to our knowledge about development, disease, and perhaps therapeutic intervention. Well, let me refer to Dr. Abyzov, who talked about somatic mutation being birthmarks that will allow us to reconstruct the cellular history of our bodies. I'll continue that discussion by making one observation that initial analysis of this cellular history actually revealed pretty interesting situations in which cells kind of compete and battle with each other. In this tug of war between one cell and the other, some cells win and some cells lose, right? And so it happens that certain cells in our body expand and others contract. And this was previously unknown before somatic mosaicism was discovered, and we learned how to use it to understand this. Let me tell you an example. There is a phenomenon called clonal hematopoiesis in the blood that happens with aging. And that's exactly what I was telling you, that among the variety of clones of groups of cells that form our blood, some become expanded and the others contract. And so this could be pretty important to understand ourselves, our histories, and our predisposition to disorders.
And I'm going to talk about the importance of collaborations for the SMaHT network. Somatic mutations are quite technically challenging to study, so it's very important to bring together scientists with diverse backgrounds and skills to work together to advance the field. And one of the most important strengths of the SMaHT network is that it brings together researchers from across the country to brainstorm new ways of detecting somatic mutations, new ways of analyzing somatic mutation data, and new ways of applying somatic mutations in order to understand human health and disease. And every research group in the SMaHT network is involved in collaborative projects, and the collaborative projects are also important for making sure that our results are as rigorous as possible, and we do that by comparing our results to each other and validating them together.
As an MPI, my role is to primarily oversee the production operations for the project. I ensure samples come in and out of the door and flow through our sequencing facility. In addition, I'm a co-chair of the benchmarking working group, where our goal there is to benchmark our variant detection and ability to find somatic variation in samples.
So first I'll break down what this means. SMaHT is really hoping to make some huge strides in terms of how we identify disease, how we understand why diseases happen in some people and not others, as well as many other scientific goals. At the core of this type of pursuit is this idea that we should be able to develop better preventative measures, better early interventions, better ways to identify diseases as they're initially happening. And really, all disease goes back to how our genes are interacting with the environment, how our physical body is interacting with the environment to produce cells that aren't doing what they're supposed to do. If we truly believe that health access equity is an important core value of who we are as a society, then doing research that fairly and equitably represents everybody has to be one of those core values as well. If we're able to do this research on a diverse, representative sample of people, and so we're able to really understand how cells are changing over time in all groups of people, then we stand to be able to really benefit everybody fairly. We stand to have outcomes of this research that has better treatment options that are tailored, that really are suited toward all of the different types of ways that cells can change over time for different people. We're really getting at the root of the issues when we're talking about health inequity in the United States and across the globe.
I believe the SMaHT Consortium has really the unique opportunity to advance precision healthcare and precision medicine by getting into unprecedented depth in cataloging somatic mutations in a variety of human tissues. We've all known since over 15 years how many inherited variants are present in all of us. It's about 4 to 5 million sequence variants per person. However, with the amount of somatic mosaicism that is created in each of our cells every year, actually every adult carries more somatic mutations in them than all of the humanity has present in their germline. And with SMaHT, we're going to understand not only where these variants are, but we're going to approach what the functional effects of these variants are, in which cell and tissue contexts they occur. And I believe this will present many opportunities to advance precision medicine, to allow for more nuanced treatments of patients based on their genetic profile, not just the germline genetic profile, but individual profiles of somatic genetics in tissues of interest that are relevant for disease. There are some examples for that already that are coming from work from colleagues in the field. We know, for example, from the clonal hematopoiesis field, that certain mutations leading to clonal hematopoiesis give a much higher prior to later suffer from cardiovascular disease than high blood pressure, which is historically seen as a strong risk factor. And we now need to go into other tissues and understand both where the variants are and what they do to advance precision medicine. And I think SMaHT is in an absolutely outstanding position to achieve that.